Yeah, so I'm happy to present to you and introduce Dr. Olivier Goureau. He is a research director at INSERM, the Institut de la Vision at the Sorbonne University in Paris. Dr. Goureau was trained at the Orsay University, and after his PhD, he studied more than 20 years different molecular and cellular mechanisms controlling retinal physiopathology at INSERM at Paris and at UCLA at Los Angeles. At 2003, he joined the research group directed by Professor Sahel and is currently heading the team Retinal Development and Repair at the Vision Institute at Paris. His team aimed at understanding retinal differentiation mechanism, at developing stem cells-based therapies for the treatment of inherited and acquired degenerative disease of the retina, where there is death of retinal ganglion cell or the photoreceptor or retinal pigment epithelium. He is also doing model of human retinal degenerative disease by generating 3D retinal organoids from patient-specific induced pluripotent stem cells. Dr. Goureau has authored and co-authored more than 90 peer-reviewed articles, and I'm happy to have you here with all the distinguished guests that we have here. So, please.
It's okay?
Yeah, it looks good.
Okay, I would like to thank the organizer of this seminar series, and especially Yossi, for inviting me to present the one part of the result of our teams. As you mentioned, we develop a lot of a new therapy for treating retinal dystrophy based on the replacement of the retinal pigment epithelium, but also the photoreceptors. And what I would like to share with you today is the one part of our study, which is more focused on the retinal organoids that we can derive from the pluripotent stem cell, and then how we can use them to understand the retinal development, and particularly to understand some specific retinal dystrophy, and to develop different strategy to manufacture the retinal cell and some mini retina in 3D that we call retinal organoids from this stem cell. Just to make a brief definition of organoids, with the general meaning being similar to an organ. Organoids are the complex 3D structures that are developed from stem cell or organ-specific cell, and they display architectures and functionality similar to in vivo organ. I will say they try to have in the minimum one functionality which is similar to the organs. And an important point is that many of them are generated through a self-organization process. And as you can see in this cartoon from my colleague in Switzerland, you have different level of maturation for the organoids, depending, they are more close to the fetal tissue or to the adult tissue. And also the big difference is the way you generate the organoids. What is the cell of origin? Then actually many people derive tumoroids. We are coming from a cancer cell. And some organs possess some adult specific stem cell that you can isolate and use them to generate organoids, as the case for intestinal organoid, for example. You can use some fetal cell for the pancreatic organoids, but many organoids are generated from pluripotent stem cell. Then what we mean by pluripotent stem cell is the human embryonic stem cell, as you know, and the iPSC, the induced pluripotent stem cell that has been discovered by the Professor Yamanaka in Japan in 2007, and he get the Nobel Prize for this amazing discovery. And as concerned the in the context of the retinal organoids, actually they are always produced from the pluripotent stem cell, from iPS or ESL. We don't have any adult stem cell in the retina that could allow us to generate the organoids. And the question is, how can we produce retinal cell or tissue as complex as the retina from the stem cell. And you see here one pictures of the retina, which is a well-laminated structures and the presence of specific cell type in different layer with the photoreceptor here that are the photosensitive cell in the retina, and the bipolar cells and the ganglion cell with the axon from the optic nerves. And then how we can try to make a retina that can recapitulate these structures very well laminated into the dish. The idea is now go back to the developmental biology by trying to mimic the development of the retina in the dish. And this is for this reason that actually then many people that are starting to make organoids are people coming from the field of developmental biology, because we have a lot of knowledge about how the retina is formed in different species, like the mouse, for example. And the idea when we start to do the organoids, to make organoids in the dish, was to try to reproduce in vitro in the dish the key step of the retinal development. And in these two slides, I just mentioned what are for us the key step of the retinal development. Then the first one is the specification of the eye field territory in the forebrain. And in anterior brain, you have one place where the cells are under the control of specific growth factor and morphogene gradient. Then you can see they have a high level of insulin growth factor, high level of fibroblast growth factors, low level of BMP, and low level of Wnt. And in this context, this different gradient promote the transcription of specific transcription factors that we call eye field transcription factors. And this cell co-express these 6 transcription factors, and they are committed into the retinal lineage. And then they are committed into the neural retinal lineage. They form the optic vesicle here, okay? And when the optic vesicle is formed, this neuroepithelium here is in contact with the surface ectoderm, and you have a process that we call invagination, where the part of the optic vesicle in contact with the surface ectoderm form the presumptive neural retina, and here forming the lens plaquette, then the future lens. And this process is still under the control of different growth factors and morphogene Wnt and sonic hedgehog, FGF and TGF beta. And finally, in this presumptive neural retina here, you have what we call in the beginning the multipotent retinal progenitor cells. And this is the cell that are able to give rise to all the retinal cell type in a very conserved chronological orders. It's the same from the cheek to the human retina, for example, with the ganglion cell that appear first, following by amacrine horizontal, cone onward photoreceptors, and finally with bipolar and Muller cells to form this laminated structure which is the neural retina, with the outer retina here, inner retina, and just in front of the neural retina, still keep in mind that we still have this pigmented epithelium, which is required for the good functioning of the retina. As you know, a dysfunctioning of the pigmented epithelium is at the base of the age-related macular degeneration, for example. Okay, then this is the key step that we have. We know about the development and how we can apply that in the dish. This cartoon is to illustrate actually the 2 major protocols used worldwide to make some retinal organoids. Then the one we developed here on the top, and the one developed by the group of Dave Gam in Wisconsin, in the Ann Arbor, in Wisconsin University. The only difference is starting from aggregate of ES or iPS cell, or in our condition, we start from adherent pluripotent stem cell. And in both cases, the idea is to put them in a specific medium, which is what we call neural induction medium, that allow the formation of these structures. And then after we pick up the structures, which is a manual step that we could discuss if you want, but that could be a little bit problematic if you want to industrialize and to make some cell for the cell therapy. And we can culture these neural structures as a floating culture in the retinal differentiation medium and the retinal maturation medium to form some retinal organoids. And just to go more into the detail in 3 slides about how we can do that in the lab, just to illustrate here, this is a colony of iPS cell, but it would be the same for the ES cell. And we are in, I can say, GMP-like condition, then we don't use any feeder and we don't use any animal product, and the medium are very well defined. Then the idea is to, when the iPS cell start to overgrow, and you see here they start to touch, then they start to differentiate. And we remove from the medium the pluripotency factor, which is the FGF2, that promote the differentiation of the iPS cell. And to favor that differentiation into the neural ectodermal lineage, we switch the medium from iPS medium to the neural injection medium. And then after less than 1 month, I would say between 3 weeks and 4 weeks, we have these structures that appear in the dish, and we can pick up the structures and cultivate them in 3D to form retinal organoid that start to mature. And how this process can happen in the dish, because we just play with cultural medium. The first one is if you try to identify this cell population by RT-PCR, what we demonstrate is that this cell express the 6 transcription factor, 6'3", LH62, MITF, RAX, PAX6, and VES62, demonstrating that the cell present in these structures are retinal progenitors with a high field territory identity. Then we find a way to recapitulate in the dish the first step of the retinal development, which is the high field territory specification. And what is interesting is because we don't block the BMP and the Wnt pathway in this condition. And we try to understand how it works. And if we look by RT-PCR about the specific expression of Nogin and DCOP, that are 2 inhibitors of the TGF-beta, BMP, and Wnt pathway, what we saw is that during the cultures, we have an endogenous production of this inhibitor, suggesting that we have an endogenous inhibition of the BMP and the Wnt pathway, meaning that we are relatively, we can mimic this condition with the FGF, the EGF, which is in the culture medium, and the Wnt and the BNP that are inhibited by the presence of the 2 transcription factors. And then if we just after leave the organoids in floating cultures, then what we can see is that we see the appearance of all the retinal cell type exactly in the same chronological order to the one we expect in the normal development. Then we have the ganglion cell that express BRN3E. We start to see the first photoreceptor precursor expressing microverin in green here. And then if you wait longer, then we can nicely see here the horizontal cells, the amacrine cell that co-express the transcription factor AP2 and PAX6, and the retinal ganglion cell here on the top. And we can also distinguish between the presumptive outer retina and inner retina, where are the photoreceptors identified with CRX, and the non-photoreceptors that express PAX6. And as you can see, we need to wait for a longer time in cultures and more than 150 days. I would say between 150 and 200, with the appearance of the bipolar cells that co-express the protein kinase C and VES62, and the Muller cells that cross the entire part of the organoids by the co-expression of these 2 markers. And if we focus on the photoreceptors, then here on the bright field, then you see that the organoids, and you see this kind of airy structure here, this filament here, correspond to the formation of the outer segment of the photoreceptors that can be identified by immunohistochemistry. Then you can see here with the marker of rod and cones, respectively rhodopsin and cone arrestin. Then we have the rods here, we have the cones, both the red, green, and the blue opsin. And I just put here a slide, which is a cross-section of the human retina post-mortem, where the guys try to identify the cones here. And then you can see that the situation we saw in the organoids is relatively similar to the normal human retina. Then suggesting that we have here a very good tool to recapitulate the development, also to make a retina in the dish. And another way to characterize the cell population is the single cell RNA-seq. Then what we did in collaboration with a group here in the Curie Institute in Paris, we collect the organoids at different stages, and I just illustrate here one stage, which is mature organoids with 2 different iPS cell lines. And we can nicely show that we have both rods and the two subpopulation of cones. We have some photoreceptors that are not fully mature, bipolar cell, a macrine cell, horizontal cell, the Muller cells and the glial cells here, few mitotic cells. And something is expected is here that we don't have any ganglion cell because the photoreceptors, the organoids are, I would say, very old. And the ganglion cell, they start to appear in first, as I mentioned before, close to 50 days after the differentiation. But since they have no way to project, because we don't have any brain target into the dish, they start to degenerate. Okay, it's like if you just remove the retina from the mouse, for example, and culture them ex vivo, then you lose the ganglion cell in 2 days. And here, actually, then some people try to develop some more sophisticated organoids where you can promote the survival of the ganglion cell by using some brain tissue, for example. Okay, then here now, this is the organoids that we have, and then how we can just try to visualize these organoids in 3D, and how we can just manipulate them to make some reporter organoids with the idea to make a specific cell fluorescent. And this two movie is just to illustrate the strategy we developed in the lab, which is to make transparent the organoids. Then the idea is to make the immunohistochemistry in 3D in the organoids, and then after make the organoids transparent and try to see the cell in the organoids. Then you can see here, this is the photoreceptors. Then on the top, this is in white, this is all the photoreceptors, and in red, this is the cones. And on the bottom, this is the cone are in green, and then you can see that they have a very nice formation of the outer segment, which can we say the body type like structures. And we have the ability to develop this kind of strategy to visualize the organoid. And if you go deeper into the detail with a high resolution microscopy, then meaning that it could be a confocal with very powerful objectives, then you can see, for example, here by making transplant organoids and after making immunohistochemistry with the rhodopsin, we can show this kind of filament here that correspond to the formation of the outer segment that we can see into the organoids. Then suggesting that you can use this strategy, for example, if you want to follow some disease where the photoreceptors are disorganized and where they should die depending of the presence of mutation. Okay, then the second step is it's okay, but how we can make some fluorescent organoids that we call frogs here from fluorescent retinal organoids. We develop a strategy which is based on the CRISPR-Cas9, where we find a site in the EVS1 locus in the human chromosome 19, where by knocking, we can add some fluorescent protein driving by a promoter of your choice. Then here, for example, this is the TFP, and the promoter is a ubiquitous promoter, meaning that normally, if it's worked, then all the cells should be green. And then you can see here the colony on the top right, the IPS colony where all the cells are green. And then if you apply or protocol of differentiation, then the organoids start to be green. And if we make some retinal section, all the cells here are green. And we use, for example, this iPS cell line to isolate the ganglion cell and to transplant them in some specific model of optic neuropathy. And these cells, for example, are very useful and if you want to make some transplantation to follow the cell. But more interesting for us is how can we generate some specific fluorescent reporter cell line that should allow us to follow the differentiation in specific lineage. And we can do that for a ganglion cell or for a photoreceptor, for example. And I just illustrate one here where still the same strategy, knocking in the AVS1 locus, the M cherry, the red protein, the red fluorescent protein, is targeted into the nucleus by a histone motif, and specifically in photoreceptor because the pilot of the M cherry is the Cerix promoter. And still by the differentiation protocol here, you can see that you have some place where a large number of green cells appear. And to be sure that these cells are photoreceptors, then you can make some section. And what you can see here on the top is that all the M-cherry cells, okay, are also recovering positive here, confirming that these cells are really photoreceptors. And this cell line is particularly useful then if you want to characterize photoreceptor cell population for the organoids, for example. That could be also used for the cell transplantation, but the problem with the cell transplantation here is that the M-cherry is, the staining is very bright and intense, and it could be problematic for the cell transplantation. Okay, then here we have the organoids, and we have the architecture, we have all the cell type, and then the main question now is, go back to my definition in the beginning of the organoids, that normally you should recapitulate one function or minimum one function of the organs. Then the main function of the retina is to catch the light. Okay, then the idea is, can we record some light response for the retinal organoids? And here, this is the phototransduction, all the protein that are involved in the phototransduction, where the main when the light catch the rhodopsin at the final, you have some hyperpolarization of the photoreceptors. And in this slide, I try to recapitulate the few papers that have been able to record some functionality into the organoids, okay, by 3 different techniques. That could be a classical electrophysiological recording with the patch clamp on the photoreceptors. That could be on the photoreceptor on the organoids, but many of them are for the photoreceptor that have been dissociated from the organoids. Some group use the multi-electrode array, which is one way to record the retinal ganglion cell, then meaning that you can just record the ganglion cell in response to the stimuli of the photoreceptors. And finally, the calcium imaging, which is also used. And this is all the paper that have been doing that, but very few of them use the light response. And we believe that actually it's maybe the problem with the light response is the fact that you need to dark adapt the cell. You need to have normally the RP, because the RP is important for the replenishment of the chromophore. This is for this reason that it's very difficult to have some light response. And the top here, on the bottom here, the paper here from Valeria Contosoler, published now more than 6 years ago, where she's the only group that have been able to record the photoreceptors, the light response by patch clamp directly in the organoids. The strategy we developed here 2 years ago is how can we mimic the dark current into the photoreceptors. Then we have not been able to record the photoreceptor directly in the organoids. Then what we did is we dissociate the organoid after 200 days in cultures, then making that you have mature photoreceptors. Then we can identify these photoreceptors with a specific antibodies, which is the CD73. I will go back on that later, which is coupled to the PE, and then this is the photoreceptor here. And we try to record the calcium by calcium imaging after a puff of cyclic GMP, which is the guys that allow the opening and closing of the channel responsible for the hyperpolarization. And what we are able to show is that these 2 responsive cells here that are red can also quench the fluorescence of the Furet2, demonstrating that you have an increase in calcium into the cell, which is one way to demonstrate that these cells are really photoreceptors, because this is the only cell population that have this ability to respond to the cyclic GMP. Okay, then this is actually all the strategy we have developed to characterize these organoids from imaging, RNA-seq, and also function. And actually then, based on these organoids, then different groups start to use them for cell therapy, for gene therapy, from pharmacological screening, or for disease modeling. And since I don't have time to go to all this strategy, I just want to mention first how we can use this organoid for the cell therapy, and particularly to replace the photoreceptors. And you have 2 ways when you have 2 ways to make the transplantation. Then you can just cut a sheet of the organoids and to transplant them into the subretinal space of different retinal degenerative rodent model. But this is relatively tricky because the photoreceptor need to contact the inner nuclear layer, but also to contact the RP. And the sheet you transplant is not only photoreceptors. Then you have some interference with different kind of cells that should be problematic for the visual recovery. But some groups try to do that, particularly the group of Masayo Takahashi in Japan, and they have some success with that. The other strategy is to dissociate the organoid, and then you can transplant all the cell, but it's the same problem. You don't have an homogeneous population of cells. And the strategy we choose, and that many groups choose, is to try to isolate a population of photoreceptors before to transplant them. And you can use the FACS, but for do that, you need to have some reporter cell line. And thinking about cell therapy in the future, then we cannot transplant GFP cell into the patient. And the other strategy is to use the magnetic activated cell sorting by trying to isolate the photoreceptor based on the expression of the cell surface specific anti-G. And then after you can transplant them, or some group try to put them in the bioscaffold to make some mature sheet of photoreceptors. And the strategy we choose in the lab is based on the expression of the CD73, which is specially expressed on the photoreceptors in the retinal organoids. And the idea is to transplant a photoreceptor precursor, and the cell, the photoreceptor before they are fully mature, with the formation of the other segment expressing opsin and rhodopsin. And then based on different experiment, we find a window between 100 and 130 days where we can pick up by max the cell of interest. Then here, this is a cartoon that illustrates how we can do that. Dissociate the organoids, make the immunochemistry with the CD73, and with the antibodies coupled with magnetic beads, we can separate between the magnet, then remove the negative fraction, and then the positive fraction is retained between the magnet, and then after you can elute the CD73 positive fraction thanks to these magnetic beads. And one way to verify that is just to evaluate the photoreceptors population that co-express CRX and recoverin. Then this is the unsorted population. Then you see that we have a lot of photoreceptors as expected, but we have a cell here that do not express CRX and recoverin, that are not photoreceptors. And if you compare the positive fraction, the negative fraction, then you see that we have a nice enrichment in double CRX recoverin positive population, which is quantified here. Meaning that in this positive fraction, we have close to 80% of the cells that are photoreceptors. And then what's going on now if you want to transplant them? And we do a preliminary experiment in the nude rats, okay, because we transplant human cell into the rodent, and nude rats, they don't have any immunosuppressing system. Then we saw the cell after 2 or 3 months. Then now we move to a degenerative model, which is a rat that carry a mutation in the rhodopsin transgene with a truncated form of the rhodopsin, the P23H, which is also found in a large number of people affected by retinitis pigmentosa. Then we transplant the cell in the subretinal space when the photoreceptors are mostly gone. We immunosuppress the animals, as I mentioned, because this is the human cell. Then if you don't immunosuppress the animals, then after 1 week, you don't see any cell. Okay, then we transplant them, and we just identify the human cells. Then the 2 strategy is to use specific antibodies that recognize only human cells. And you can see here the cell, human cells. And the other strategy is to transplant a cell from retinal organisms derived from the iPS from a male, and you transplant in the female rat, and with the M-fish chromosome staining, then you can also identify the male cells that correspond to the one you transplant. Then the cells are still there after 10 weeks post-transplantation, thanks to the immunosuppression. And if we look about the characterization of this cell, because we transplant a non-mature photoreceptor, are they able to mature, to connect the retina, and finally to restore the vision? Then what we demonstrate here on the top, you see that the cell we transplant, this is the human cell here, they start to express cone arrestin, suggesting that they are cones, and few of them express rhodopsin, and demonstrating that we have also rods, but majority is cone, which is particularly interesting because thinking about the cell therapy for the patient, then the idea will be more to replace the cone, which is the photoreceptor that you use all the time for the day life. An important point is that the cell we transplant and the cone here are they express in connection with the bipolar cell of the rat, the marker of the ribbon synapse, which is the synapse between the photoreceptors and the bipolar cells. And you can see here one human cell which is in connection with one bipolar cells of the rat, that meaning that we can make some synapse activity. And this slide is just to resume the positive things we get. It's just we have one way to isolate photoreceptors, an homogeneous population. All the process, the differentiation and the selection protocol are ready to transfer to the fully GMP condition. We demonstrate that the cell we transplant can survive in the degenerate retina, and they can be differentiated mostly into the cone. And then the question now is, can we able to restore the vision? And the answer is no. Then if we try to record the electric activity of the retina by the electroretinogram, we don't see any difference between the control and the transplant cells. And then one explanation may be that when you record the ERG, you record the full activity of the retina. And when you transplant the cell, you only save one part of the retina. And then maybe the part you save is not enough to see any global ERG restoration. But the major issue with the photoreceptors transplantation is that when you transplant the cell, then we demonstrate that they can connect to the inner retina, but they don't form any other segment. The photoreceptor don't form any other segment. Then are they able to make a functional phototransduction? We believe that not. And also that they don't have other segment and they don't connect with the RPE, meaning that the contact with the RPE is required for the chromophore replenishment in normal phototransduction. Then the two strategies to try to tackle this major issue is, the first one is I briefly mentioned before, is to make some scaffold to promote the maturation and the polarization of the photoreceptors. Then you isolate them, you make the scaffold, and then after you transplant the cell into the scaffold. And the second strategy is the one we start to develop here in the lab with the group of Denis Dalcara and Jens Tubel, is maybe we can just artificially confer the light sensitivity to the cell we want to transplant by combining gene and cell therapy. And how we can confer the light sensitivity to the cell. Then this cartoon here just show all the actors of the phototransduction that are present in the other segment, leading to the hyperpolarization. That is the first signal to send the signal after to the bipolar and the ganglion cell. But in our case, we have this kind of photoreceptors that never form the other segment. Then how we can recapitulate this kind of strategy, the hyperpolarization. And to do that, we plan to express in our cell an optogene, microbial opsin named Joe, which respond to the orange light and open after the response to the light, allowing to enter the chloride ion, mimicking the hyperdepolarization. And then the optogenetics has been used for different strategies to restore the vision. And I just want to try to recapitulate here the different strategies. Then you have the normal retina here, the RPE, the other segments. And for the optogenetic, actually, the approach is by the gene therapy. Then you have some adenoid virus that allow to express the microbial opsin either in the remaining cone or in the second order neurons, the bipolar cells. Then in the first case, the first situation, the recovery is dependent of the viability of the remaining cone. And for targeting the bipolar cells, this approach loses the information processing of the inner retina due to the absence of the cones. And in the cell therapy, as I mentioned before, the cell we transplant, they don't respond to the light. Then the idea was to how we can combine these two approaches here, the optogenetic and the cell therapy. The idea is try to express a microbial opsin into the photoreceptor derived from the pluripotent stem cell before the transplantation, making this cell sensitive to the light, and then after transplanting them. And they are normally independent of the formation of the other segment. They don't need the RP for the chromophore replenishment because they have the microbial opsin, and they should transfer the information to the remaining bipolar cells. And what we did is just we make organoids from the iPS. We transduce the organoids with adenovirus that express this hyperpolarizing chloride pump named JO, which is a redshift allorhodopsin, which is derived from the Allobacterium salinarium. And what we have been able to do by patch clamp here directly on the organoids after transduction is here you can see some photocurrent response with 2 consecutive flash of light. And when the cells are transduced only with the GFP, no JO, we don't have any response, then demonstrating the specificity of the response. And here this is the action spectrum of the JO cone stimulated at different wavelengths. And we have the maximal response as expected for the orange light. And then what's going on if we transplant into the mice? Then if we transplant into the mice, you see the cell here that express GFP in the contact of the bipolar cells. Then we can record the photocurrent elicited by JO in the donor cells, but also in the second order of neurons, here the bipolar cells. And more demonstrating that when the photoreceptors are sensitive to the light, and they can transmit the information to the bipolar, and they can also transmit the information to the ganglion cell, because if we use this multi-electrode array here, where the retina is ex vivo, is flat onto the different electrode here, when we flashed with different light, with the light here, you can see that we have both on-off response from the RGC, which is specific for the presence of JO, because we don't have any response to the GFP. Okay, then demonstrating that we have a one way here to restore the vision after transplantation of the photoreceptors that have been artificially sensitive to the light. And just to conclude on that, where we are today is we try to move to the preclinical study, to the non-human primate with the macaque, where we have the advantage here to have a big non-human primate facility with the surgeon dedicated on that. And the idea is to use both iPS from macaque and from human to combine this approach of cell therapy, and also to make some allograft to have some compatibility between the iPS donor and iPS receiver in the macaque, which is reflect more the strategy that could be used in the patient, and try to avoid the xenografts that need to immunosuppressive regimen because you transplant human cell into the macaque. Okay, and the last 5 or 6 slides, which is one that have been already engineered to shift to the red allorhodopsin, meaning that she need less light intensity to the classical allorhodopsin.
Okay, thank you very much. Any more questions?
Yeah, maybe if I can ask. Hi, first of all, thank you for the talk. Really, really interesting results. My question is rather about missing cells that you were also pointing out at the very end. So one cell type that usually is missing, and it's quite crucial, I would say, is the microglia cell type. And this also in brain organoids, for example. But of course, for disease modeling as well as for the development of the retinal structures, that are crucial, really, cell types. So are you planning on implementing them, for example?
Actually, not really, because it's a different group that are doing that, and then we cannot do all. And just I know that my colleague in Australia, they try to doing that, and it's not for the microglia. I would say will be not too much difficult, because actually you have a lot of commercial microglial cell derived from the iPS, and you have some people that try to use some hematopoietic stem cell that could give rise to microglia, but we don't do that in the lab. But you're right, it will be interesting. Then I know that for the brain organoids, actually it's start to see some some paper where the people add directly the microglia into the organoids. I would say it would be more, it would be easier compared to make some vessel to vascularize the retinal organoids. Because if you look at the retina on flat mounts, then you have a lot of microglia in the inner layer, but also very few at the level of the photoreceptors that just came when you have some inflammation. Then I would say that maybe just add at one moment the microglia cells derived from the IPS into the organoid and to see what's going on.
Right. Thank you.
Welcome.
Okay, thank you. Any more questions? So, well, if that is the case, then we are just on time. So let's thank you again. It was really a great talk.
Thanks, Yossi.
Interesting. Thank you very much.
Thank you very much for the invitation.
We are looking forward to see more progress, maybe next year.
Yes, we hope. But with the you know that actually it's like a pandemic worldwide, and I know that in Israel you are lucky because now all the bar is open and everybody is vaccinated. But here in France it's very complicated, particularly for the animalry, and then for the macaque it's a mess. But we hope.
Yeah.
Thanks.
Okay, I hope so. Thank you very much.
Thank you very much.
Thank you.
Thank you all.
Bye.
Bye-bye.
Okay. Bye-bye.